Good afternoon, everyone. My name is Matt Berriman from the Sanger Institute. And my group's been working on the genomes of NTD parasites for several years. We've got a particular focus on parasitic worms, and today I'd like to briefly dive into our genomics work on schistosomes. The objectives of our work are really twofold. Firstly, to understand the genomic basis of parasite biology, and secondly, to investigate parasite genome evolution and demographic changes over different timescales. Our hope is that the genome biology part will expose new vulnerabilities to be exploited in parasite control, and that the genome variation part will reveal, amongst other things, changes to parasite populations in response to control measures. These two areas are clearly dependent. Our knowledge of the genome powers our ability to study variation, and identifying signatures of possible adaptations can direct our attention to specific parts of the genome that we then need to dive in to explore the functional consequences. I'm going to really focus today on the genome biology part of our work, but I'd firstly like to quickly recap on some of the work you may have heard at previous meetings from colleagues in my team. To study parasite populations, we need a method to collect the parasites, of course, and existing collection methods used by others in the field for many years actually work quite well for this. Parasites can be recovered from stool, filtered and washed, and then placed on FTA cards. The work is obviously very labor intensive, but if the parasites are washed carefully, you can actually recover DNA that is good enough for whole genome sequencing without loads of contaminating bacteria in the way. Here we see the results of Duncan Berger's analysis of data from parasites originally collected and sequenced by Tom Crellin. Over 200 parasites were compared from schools from 2 districts in Uganda that differed in their MDA histories. From about 4.5 million SNPs, putative signals of positive selection were calculated based on genetic differentiation, bottom panel, and 2 methods for looking at conserved regions of the genome that have not been broken up by recombination. The distance between these 2 districts is a potential confounder, but it is at least tempting to speculate that these signals that we're seeing are actually positive selection due to differences in MDA pressure. Going from loci in the genome to their function is really hard. We clearly need the underlying reference sequence and gene predictions to be as accurate as possible. And to improve the reference genome, long-range information is absolutely critical. You get that either from long reads or from various physical linkage mapping approaches. You can see the effect of folding in this kind of information into the S-Mansoni genome here. In the latest version on the right-hand side, all of the smaller pieces that were previously present are now joined into the main chromosomes. And this includes the sex chromosomes. So why do we care about the sex chromosomes? Well, schistosomes are dioecious, which makes them unusual amongst flatworms. But the factors and processes that determine sexual development in schistosomes are unknown and could make excellent targets for developing new interventions that would essentially block transmission and block pathology. To make headway in this area, we clearly need to understand the structure of the sex chromosomes. The Z chromosome comprises pseudo-autosomal regions at either end that have homologous regions on W. Between them is a Z-specific region that can be clearly seen when we map sequence data from other females, which is ZW, to this reference. About 1/3 of the chromosome is DNA that is essentially unique to Z and is therefore single copy in females rather than 2 copies. Across this region, there are about 900 genes, and any or all of these could be important for sexual development. The W chromosome has been much harder to piece together. Its sequence is degenerate and repetitive, but threaded amongst these blocks of repeats seen in black and gray at the bottom of the diagram there are about 30 genes which are called gametologs, which is a name for a gene where the homologs are found in the Z-specific and W-specific regions. And in many of the cases for these gametologs, the sequences are really quite diverged. So again, they could have a role in sexual differentiation. Improvements in sequencing methodology mean that we can start to apply long read approaches to more diverse samples. Here we can see a summary of Duncan's progress in sequencing across the genus using samples from SCAN and several other collaborators. In many cases, libraries have been made from as little as 1 to 3 worms. And if you look at the assembly metrics, the total genome size and contiguity are on a par with existing published reference genomes. So the genomes are coming out at roughly 400 megabases, and we're getting scaffolds with N50s of roughly 50 megabases. We've only just begun to look through these data, but can already see some major structural variations across the genome, including rearrangements within Z that actually do not correspond to the boundaries of events that we saw in S. mansoni. So in the bottom right panel there, you can see this big inversion in schistosoma spindali, and then several other inversions going into intercalatum and hematobium. I've rattled through this quite quickly, but I just want to summarize that I've talked about how individual larvae can now be used to collect several million high quality SNPs across the genome from individual parasites. I've also talked about the impact long read technology is having. It's enabling us to really complete the job of producing chromosomes, and from that new information, we're beginning to be able to investigate the genomic basis of sex in schistosomes. I've also hopefully given you a taste of things to come in terms of how we are trying to understand how the genome has been shaped over a longer time scale, and how we can use that information to direct our attention to important areas of the genome. Very quickly, just need to acknowledge some people, lots of people have been involved in this work, but from Sanger, I must point out Duncan, Sarah, James and Alan heavily involved in all of this sequencing work. The SCAN resource has been amazing for supplying population genomic samples and samples for the broader pan-schistosome analysis. And I'd also like to gratefully acknowledge that we've been welcomed into several long-term collaborations led by Joanne Webster, Alison Elliott and Emily Webb, involving lots of partners in the Ministry of Health and the MRCU VERI in Uganda. Thank you.